各位老师，那各位同学，各位同好，大家好哈。那今天非常高兴能够邀请到何雅琪老师、医师、博士，对，大师来给我们分享跟演讲。那雅琪这一次的机票当然不是我们出的啦，不过我们知道她回来，我们就赶快把她抓来这里一起做这个演讲。那雅琪跟我们这个的渊源其实蛮深的，我们这个系列的演讲在疫情期间第一次可以全线上的演讲，就是由雅琪在帮我们演讲的，所以目前我们这个演讲hybrid 的，就现场跟线上这个部分归功于雅琪当初的好演讲。那我来介绍一下何雅琪老师哈。就是何雅琪老师目前是在耶鲁Yale 当助理副教授，然后他在的部分是在Department of Microbial Pathogenesis 然后他同时也是内科的合并的这样子的副教授。那他在Yale的也还负责Microbiology Department 的招生，好，所以如果大家自己或者是有知道好的学生想到美国，然后有好的机会，这个我们是有很好的一个切入点。那何雅琪老师他成大医学系毕业他一路就是拿一堆奖项比如说成大医学系毕业就是斐陶斐啦，然后到台大当内科医师、内科的住院医师，还有感染科的 Fellow 的 training， 也就是这种最顶尖的住院医师也是拿一堆奖项。然后他临床医学研究所可能是 R4、R5 拿的一个硕士，然后主治医师云林待了1年，然后就到美国 Johns Hopkins 的拿 PhD 那个时候就开始做 HIV。然后他在美国拿 PhD 的时候也是拿一堆奖项然后包括Howard Hughes 的International Investigator， 就是 Young Investigator。然后他在 Hopkins 拿了 PhD， 做了 Postdoc， 那也在 Hopkins 做到 Faculty 的 Track， 但是之后他就去了耶鲁。那在 Yale 他就是Assistant Professor、Associate Professor。那也拿了一堆 grant， 拿了一堆奖项，然后他的研究的主轴当然 HIV 一直是他的最爱，然后但是在做 HIV 的时候，他同时也接触了许多或发展了许多的新的技术包括目前的 single cell 等级，然后 Spatial 就是空间等级，然后 multiomics 的一起的 approach 来探讨为什么某一些 HIV会躲起来为什么某一些杀不掉，那怎么样去找他们。那他的 papers 是多到不得了，那在 Cell， 在 Nature Medicine、PNAS， 然后 Immunity 等等很多很多，然后他的技术可能也都走在很前面的地方，所以大家如果有一些技术的发展或者是想法上的讨论，都非常值得跟今天的这个演讲或者是以后的长期的联络。那我们欢迎雅琪。对，
谢谢，这是非常高兴回来分享一些我们最近在做的东西，然后真的很开心可以跟大家讨论一下。那就是我之前就是在这边长大，那我有学一些新东西，看大家可能刚好有一点兴趣，我们可以继续讨论。那我可能讲还是用英文，因为不然有些专有名词可能不是很好翻译这样子。So the focus of our research is to try to understand HIV persistence at the single cell level, and the reason is that because we want to understand the heterogeneity of biology. So the biological heterogeneity is still the major key unsolved puzzles in medicine. The reason is that when we're thinking about all these difficult diseases that we're treating, that we have difficulties treating with, such as cancer, The problem with cancer, for example, is that people are diagnosed with cancer, you give them therapeutic strategies, but somehow they can just develop drug resistance, they can recur. And why is that? And second is at an infectious disease level that all these infections may have different pathogens that are evolving in different populations, that this heterogeneity making treatment more difficult. So the way we see this problem is that there are three dimensions that we need to think about. First is over time. Over time, when the development of a disease toward the full-blown diseases, during time, what happened? And second, what happened at the different locations in space? For example, in the infectious disease part, then we'll see these microbes may form biofilms, and the bacteria may behave very differently in this biofilm formation. And then second, in the context of cancer, these cancer cells may develop into different clones, which have different proliferation capacities of drug-resistant profiles. And finally, we want to look at the dynamics of these cells, such as cancer, for example, if they can survive, they proliferate. And when we have ongoing immune pressure or drug pressure on them, they'll further evolve. And all these evolution and dynamics of proliferation will lead to uncontrolled infection or tumor growth. And therefore, this is the reason why we want to use single-cell approaches to understand biological heterogeneity. So in our context, we like to study HIV, although HIV can be treated with antiretroviral therapy that reaches a clinically undetectable viral load that currently all HIV-infected people can live quite a normal life. The problem is that despite antiretroviral therapy of 30 years, that once the antiretroviral therapy stops, the virus will come back. And why is that? This is because HIV resides in these infected cells called the latent reservoir, which means that they are HIV-infected cells that are having HIV integrated into the provirus, into the genome, so that you can never get rid of them. And these HIV-infected cells can persist lifetime. So therefore, because they persist lifetime in the cell, so your plasma viral load is 0, but they're inside these cells that they're persisting, and that's why they can persist lifelong. So despite all these antiretroviral therapy, ART doesn't cure HIV infection, because if you think about how ART works, that there are these infected cells. Although plasma viral load is 0, but these cells don't die. Especially ART is actually not cytotoxic. You can take your cells and throw all the ART in and they don't die because they're well tolerated. And second is that the ART does not really suppress HIV promoter. HIV is now a provirus in the genome, so then there's a promoter driving the gene expression. Antiretroviral therapy are just compounds inhibiting viral enzyme function, but it does not suppress the promoter function. And therefore, despite we can suppress these ongoing viral replications in the plasma, these latent infected cells will still persist. And then the ongoing transcription driven by HIV promoter may still drive active HIV protein or RNA expression that leads to chronic immune activation. This is pretty much like the dandelion scenario that although you don't see any dandelions in your backyard, then they can still come out upon stochastic activations such as antigen stimulation, or in the nature context, such as rain or sunshine. The current problem is that not only they persist lifetime, but these HIV-infected cells actually proliferate. So our challenge is just not these cells staying there for 70 years. Instead, these are cells that are proliferating from 1 into 2 into many, many. And this is why, especially in the United States, people do care about HIV cure, because although we have this effective antiretroviral therapy, people having HIV are still having chronic immune activation. There's a still constant cost to cover people having HIV infection for lifelong, and there's still quite some stigma against these people living with HIV. So this is what we had before, that we see that throughout these years of research, we found that HIV-infected cells are actually very heterogeneous. That this biological heterogeneity is the problem that we're faced with, because, so first, HIV integration site is very heterogeneous. They can integrate into any genes, including about 10% in the introns of, about 90% introns of actively transcribed gene and about 10% in heterochromatin. And these different integration sites drive the differences of the behavior of these infected cells. And second, HIV prevalence can be very heterogeneous, about 1% having intact HIV, but the other 99% of HIV infected cells having these provirus, having different kinds of defectiveness in the genome, but these may also cause immune activation. And third, HIV RNA expression is actually stochastic in one sense. So it means stochasticity means that these are driven by antigen stimulation and the TAR and TAT feedback loop that creates the stochasticity. Basically, they can be on today, off tomorrow, on again next week. That because this is just stochasticity. So with these several layers of heterogeneity, making us think that how can we develop an HIV cure strategy that is effective? For example, in the context of cancer, you want to know, for example, such as breast cancer, you want to know all these breast cancer genes so that you can target them specifically, such as HER2, right? But if we do not understand the heterogeneity of HIV, how are we going to design the effective strategy to target them? So we feel like right now it's not a point that we think that we know everything, we can just treat them. The answer is no, we still know so little. The current strategy in the HIV field, for example, trying to cure HIV infection is to see, okay, we just need to eradicate these HIV-infected cells. One way to do this is to use the shock and kill strategy. For example, supposedly if these HIV-infected cells are latent, they don't express any viral antigen. Now we can use these latency reversing agents to reverse HIV latency called the shock. Then these cells, when they're expressing viral antigens, they can be seen by immune responses or they can just die of viral cytopathic effects. The problem is that these HIV-infected cells, despite reactivation, having viral cytopathic genes expression, they don't die. Why don't they die? And the second question that these cells should be eliminated by cytotoxic CD4 T cells, but sometimes they survive. And why? And third, they not only just stay there, they proliferate from one into many. And this sounds the scariest part of this HIV persistence because they are just growing into many. And how do we stop this proliferation process? And fourth, these cells are so heterogeneous. So then how are we going to know enough about them so that we can develop therapeutic strategies? And this is why we are trying to use single-cell multiomic approaches to answer this question. For us, to understand HIV persistence in the clinical setting, in clinical samples, has this 1 in a million problem, that only 1 in a million CD4 T cells contain this HIV infected cell that can express viral antigens and then complete this viral cycle of being infectious. So there's 1 in a million of them that are causing this viral rebound that we want to study. However, these cells are actually hard to find. So first thing, this 1 in a million is like a needle in the haystack. It's so hard to find them. And second, HIV-infected cells actually do not have a surface marker that can distinguish them. For example, if you study breast cancer, you can take the cancer out and study them. But for HIV, they're just floating in the blood, in the body. We cannot get them out. So it's like a needle in the needle stack. And third, these cells that are infected with HIV are actually quite heterogeneous. For example, when HIV infects CD4 T cells, they can be Th1, Th17, they can be naive or central effector memory, they can be activated or exhausted, having all different kinds of antigen specificity. So trying to understand how HIV persists in a clinical setting is like trying to find this black hole in the middle of the galaxy. And this is why we want to use single-cell multiomic approaches trying to understand HIV-infected cells. So instead of trying to look at all the cells drawn, you can draw blood from people having HIV and try to study them, which is basically studying 99% of uninfected cells. We hope to tally down all these heterogeneous CD4 T cells and find that rare HIV-infected cells and trying to understand them. And so I hope that by the end of it today, what I'm showing you is that, you're doing single-cell multiomics because it sounds fancy and everyone is doing it. Everyone has this in their paper, so I sort of have to do it. Or identify signatures. The answer is no. This is not the reason why we want to do single cells. The reason why we want to do single cell is because of the heterogeneity and rarity of the biological question that we are looking at, and therefore we hope to use this method to find the biological insights so that we can validate using authentic orthogonal approaches. In the past, our field has been trying to identify these markers for HIV-infected cells, and everyone has their favorite one, including ourselves. But, now that with genome-wide profiling, which means that you can profile 20,000 genes in humans using these genome-wide transcriptomic approaches, hopefully we can try to understand these HIV-infected cells in a less biased manner. The first way to think about this is easy. So can you find those HIV-infected cells and just sort them out and just studying them? Because otherwise, 1 in a million means that you need to study 1 million cells. Great. indeed, that's what we started with. So, like in 2020, we developed these methods to activate HIV from latency and then using HIV RNA expression as a surrogate to sort out these HIV-infected cells. Same as other groups trying to use HIV amyloid expression or get protein HIV infection RNA expression as a surrogate to find these HIV-infected cells for advanced profiling. The problem is because lots of cells are just latent. So that to do these, we need to activate these cells extensively using these PMI-namycin, PHA, or CD28. And by the time you capture the transcriptome, they don't really reflect what they really are in vivo. So that, well, this is great, but we hope to know more about how they persist in their in vivo state. So the past year has been very exciting that there are several groups trying different methods to identify HIV-infected cells in their native state without ex vivo activation. So this is Ian Clark in Light Boritz, and this is Wei Sun and Matthias Lichterfeld lab, and Vincent Wu in Michael Bett's lab, and we have two methods that I want to share with you about how we try to understand this. We hope to try to understand HIV-infected cells in different layers, from DNA to RNA to protein, which is the central dogma of molecular biology. If we're able to understand not just the transcriptome, which is indeed helpful to see the differentiation status of the CD4 T cells, we also hope to identify the transcription factors that regulate this gene expression and also downstream protein expression so that we can identify therapeutic markers or therapeutic targets. So there are two studies that I want to show you with, led by my grad student, Jack Laura, and this is postdoc Yulong Wei. So to answer this question, we collaborated with Dr. Andrew to let a clinical cohort. This is a cohort of people at the village in Peru at risk of HIV infection. So what happened is that these people got, prospective HIV testing every month. You're negative, whoops, positive. Then, and they will donate blood to the clinical team, and then Dr. Zhu will save this blood during viremia. 10 to the 6 logs of the viral load in the blood. Then these people will receive antiretroviral therapy for free because of joining this trial, so that after 1 full year of antiretroviral therapy, when their viral load is undetectable, documented, then we can collect blood samples again. So from the same people, you can track them down during viremia and after viral suppression, so that we can try to understand what happened to their immune system and to their HIV-infected cells over this period of time. To do that, this is Jack Laura. This is what we reported earlier in the seminar 2 years ago, that we can try to understand the different aspects of this HIV-infected cells using single-cell multiomics. This method is called XIC. Just to know that every institute can do this. This is a standardized platform and standardized protocol everyone should be able to do. So that using XIC, the trick of single-cell transcriptomics, you just need to put 3 things together. You put your cells, your barcode, and your reagents. That's it. The reason why 10x is dominated the field is that their incorporation efficiency is about 80-90%, and that's why it seems to be more robust than currently other platforms, and that's why it's dominating the field. So when you put these things together in the bubble, so you can see this single-cell transcriptomics, because we can capture RNA, you can see these cells are Th1, Th17, or Th2. By surface protein staining, you can have these surface proteins stained with antibodies such as CD3, CD4, CDA, CD45RA, or RO. Basically, instead of FITC or PE, these 4 tags you typically use, you can have a DNA tag. So then by sequencing, you can see which cell is expressing which protein for us to dissect with their central effector memory. We can identify HIV RNA positive cells so that we can identify our favorite HIV infected cells. Then finally, we can identify T-cell receptor sequencing. T-cell receptor is very interesting because every CD4 T-cell has a unique T-cell receptor. If you capture different CD4 T-cells having the same T-cell receptor, it has to come from proliferation. So because we didn't treat these cells with anything, we just draw the blood and put them into a machine. So that if you see many different CD4 T-cells having the same T-cell receptor, it means that these are cells that proliferated in the vivo over time. So these are cells that love to proliferate, as you can see in these dark-colored cells, meaning proliferated T-cells in the body. Then because you have paired transcriptome, you can know, oh, now I see these proliferated cells are actually these cytotoxic CD4 T cells expressing granzyme be? So, all right, cytotoxic T cells are great soldiers in the body. They love to proliferate upon antigen stimulation. That's great. But when our student Jack did differential gene expression analysis, which means that if you compare HIV positive cells versus HIV negative cells, he found that, do you find markers that can distinguish them? Yeah, there are some, such as activation markers, which has been known in the field, exhaustion markers, such as PD1, CTLA4, LAC3, and TIGIT, which is also known in the field. But he found that HIV positive cells express cytotoxic T cell genes, such as perforin, granzyme K, granzyme A, so that when you look at those cells that proliferated a lot in the body, you know, those big T cell clones that has HIV in there, he found that these are the cells, that 75% of these cells having HIV and proliferated are cytotoxic CD4 T cells. So he used a machine learning, this is a random forest-based method to identify that these cells, these genes are indeed highly expressed in HIV positive T cell clones, and we use flow cytometry to validate this result, both in the typical cohort, which we identify these participants, and the independent cohort that we recruited in Philadelphia. So what does it mean and why is that? So I gave this talk about 2 years ago at Croy, and I have no idea why we find these findings. And my friend Brad Jones actually called me before just at the end of my talk. He said, Yachi, did you check Sterbing B9? I said, I have no idea what you're talking about. So turn out these cytotoxic CD4s, cytotoxic CDAs, NK cells, and even Tregs, their job is to make these lipid granules, such as granzyme B, perforin, and then to kill the target cells, such as cancer. But these lipid granules may fall back to themselves and kill them, and you don't want that to happen. So all of these cytotoxic cells may just automatically express Serpin B9, which is a granzyme B inhibitor, so that they don't get killed by themselves. It happens to be that HIV positive cells have higher level of Serpin B9 expression. And why is that? It means that HIV can infect any CD4 T cells as long as you know you have CD4 and CCR5 receptor. However, when they happen to infect cytotoxic CD4 T cells and cytotoxic CD8 T cells want to kill them, there's a problem. You have the same weapon, I have the same weapon. You have the shield, I have the same shield. So when CD8 T cells want to kill them, you may not. They may resist CD8 T cell killing. And when antigen stimulation continues, these are the cells that are normally most robust in proliferating, and that's why these HIV-infected cells can proliferate over time. So this is one reason we think HIV may persist. And interestingly, when we published this study in March, then there are 2 follow-up studies. So this is September by Michelle Nussenzweig, and then 1 year later by Rusmi Thomas. They show the same thing. So there are different groups of people identifying cytotoxic CD4 T cells as a major reservoir, just validating our study. And we're very excited to see other studies. Then people ask us, oh, I got it. You use HIV RNA expression in your single cell data to find those HIV infected cells. Yes. How about those HIV latently infected cells that have HIV DNA in them, but there's no RNA? Can you capture them? I said, no, because if you have HIV RNA expression, we capture them. But if you're DNA positive RNA not expressing, I don't know because I didn't measure them. So can you capture those latently infected cells? With that, we want to try a different method to answer this question. We use the same cohort, get a blood from during viremia and after viral suppression from the same cohort, but now we want to use this method called dogma-seq. So this is a method by Peter Smibert and Aviv Regev, initially published in bioRxiv, and we saw that later published in Nature Biotech as dogma-seq. There is a paired paper called T-seq, T-E-A, T-seq, that was published at the same time in eLife. Basically the same thing, same principle. And you can use either one, but it's quite robust in our hands. So using dogma-seq, what's this fancy term? Basically, in the past, I told you we can capture RNA-seq and protein expression together. That's what we did. But dogma-seq allow you to couple all of these with ATAC-seq. You're like, so what's ATAC-seq? So ATAC-seq basically is to use these TNF5 transposase into the cells, so then they like to bind to DNA. So that initially was designed to identify those genomic regions that are open in chromatin accessibility, that have increased transcription factor binding activity, so that you can understand the epigenetic regulators such as transcription factor binding activity that regulates your transcriptome. However, if these transposons happen to bind to HIV, then I can find HIV infected cells having HIV DNA in them. Then now, because I also understand in these cells which ones are HIV RNA positive or not, then I can identify latent versus active HIV expression. So using these fancy methods, now you have DA-taxi, RNA-seq, and surface protein. You typically see these beautifully fancy plot. You don't know what it means. They're like TH1 here and here and here, and proliferation there and there. It's just like, what are all of these? The good news is Raku Satija used this weighted nearest neighbor method, commercially available, that regularly available, that we can now integrate A-taxi, RNA-seq, and protein expression all together in the same plot. It means that every single dot you see here, we know their transcription factor regulators, we know their transcriptome and their protein expression connecting DNA, RNA to protein all together within the same single cells, so that we can identify HIV DNA provider landscape and HIV RNA provider landscape to see what are the cells that we captured. So this is the first time in my field that the transcriptional landscape of HIV infected cells can be captured at the single cell levels. This is the very first study showing this. So with that, we can capture HIV latently infected cells, which means that they have HIV DNA in there but no RNA, or actively transcribing cells, which means that they are expressing HIV RNA, both during viremia and after viral suppression. So that we can look at different aspects such as transcription regulators and RNA-seq. So I will want to just highlight the RNA-seq part because, now you can look at latently infected cells. In the past, we thought that these must be cells that just looking at uninfected cells because they are not expressing HIV RNA, they're not expressing HIV genome, so they should be as quiet and the same as uninfected cells. But from differential gene expression analysis, we found that, actually, there's some gene expression that is different from uninfected cells, and same as those transcription active HIV infected cells. So then we would like to try to compare all these differences by focusing on the genes that are shared between latent and transcription active HIV infected cells. There are many genes you can validate, but what if there are genes that are shared between these two? Maybe we should start from them. So these are genes that we sort of know about. For example, if you have HIV, you upregulate type 1 interferon response. Makes sense. If you have HIV, you upregulate cytotoxic T cell responses. We found before. Makes sense. You may have increased ALK2 production and integrate alpha-4 expression, which has been previously reported in literature. What's surprising to us is that, there's this gene called IKZF3. We don't know what it is. So turn out it's an ichorosine finger transcription factor that's regulating lymphocyte regulation, typically in B cells. That's what was known. Then after we read into literature, it turned out that, IKZF3 also help Th17 and TFH differentiation. And if you have an active domain like transcription active mutant, then you can actually increase BCL2 expression and increase cell survival. It's interesting because these are different papers that are actually seen in recently, just this year and last year, showing these. So we wonder, there is the gene IKZF3 that is upregulated in latently infected cells and actively infected cells. What does it mean? So at the same time, our postdoc did a separate analysis. This is WGCNA, a method to identify genes that are specifically expressed in HIV positive cells. So his question is, HIV RNA positive cells should die of viral cytopathic effect, but what makes them survive? For example, here, there's this gene called Ki67 that promotes cell proliferation, and there's this gene called survivin that helps cells to survive. So using WGCNA, he identified this module, which means that these are genes that are co-expressed together. So being co-expressed together, you see, for example, IL-21, IKZF3. So it means that if your cells having IKZF3, you may also express Ki67, a proliferation marker. And what is it? So the reason why we have dogma-C is that now you can see everything together because transcription factor activity. we found that there are these transcription factors such as GFOS, IRF7, and NFAC3 that during viremia are increased activity binding to IKZF3 that increases IKZF3 RNA expression. And these are the genes that are activated during HIV infection. So basically, HIV infection will upregulate IKZF3 both at the transcription factor level and at RNA level. So that we did a validation study. This is led by our postgrad Timmy. That you see these transcriptomes and want to validate what the signature you saw is true or not. It's easy. You take CD4 T cells, infect them with HIV or some clinical isolate, and try to see if you find HIV positive or negative cells. Do HIV positive cells really have increased IKZF3 expression? The answer is yes. So what's the benefit of having this? The answer is that if you're HIV infected, you should die of viral cytopathic effect. But if you upregulate IKZF3, you may also upregulate Ki67 to help you to proliferate. So we suspect that it's because of HIV-inducing IKZF3 expression that help these cells to survive. But then we did other protein profiling that I'm not going to talk about. But here is the power of why we want to understand these HIV-infected cells. So HIV-infected cells are very heterogeneous. Yes, but are there some shared biological processes or immune responses that make these HIV-infected cells, similar or that we can target on? So using ataxia-defining cell differentiation trajectory, that we found that there are different cell fate of these HIV-infected cells. During acute viremia, the HIV infection will induce interferon responses. Normal. This is what we typically know. As you can see at the ATAC level, step 1, and these IRFs. At RNA level, you express BERT5 and Ki67, and at protein level, you see these activations. So basically, during acute viremia, some cells shaped by these interferon responses will be activated and proliferate. Some cells will die of viral cytopathic effect. Some cells will develop into cytotoxic CD4 T cells driven by eons, like these. And some cells may persist shaped by GFOS that will express TNF responses and start to migrate to somewhere that we don't know. But we found that because of these multi-layer of data, now we know the cell fate decision of these heterogeneous HIV-infected cells. So that was exciting. Then people ask us, you've been studying HIV in the blood. Have you looked at tissues? And do you care about tissues? The answer is yes. This earlier study by Jake Assey showing that after ART, 98% of HIV-infected cells actually hide in the gut, in the gastrointestinal tract, in the gut-associated lymphoid tissues. Do you want to study them? Yeah, but sometimes hard. You can draw blood easily, but to do these, it sounds not very easy. But indeed, previous literature showed us that it's really these gut-associated lymphoid tissue, CD4 T cells, that are causing AIDS pathogenesis, and they're important. And those are where HIV hides. And if you look, it's not just gall. It's where there are different CD4 T cells in the tissues. They can be in the epithelial layer at the mucosal border. They can be in the lamina propria, and they can be in the secondary lymphoid tissues that you can see in normal colon, that these CD4 T cells do exist in the gut. And this is a dynamic, interesting process, and this is what we're actively studying. So what are these CD4 T cells in the gut? Gut is a mucosal interface that you may see different pathogens. If you see an intracellular pathogen, such as viruses, so we will activate Th1 because that will recruit macrophages, and altogether the job of these cells is to kill intracellular pathogens. But if you have extracellular pathogens, there's a lot of them in the gut, such as Salmonella, you may want to upregulate Th17 programs, and because Th17 will recruit neutrophils, and the IL-17 secreted by Th17 will seal the tight junction by increasing the Clutin expression. But if you have commensals, just hush, don't feel inflammatory. Don't induce these responses that they will just hush down these inflammations, so that our gut is not always in these inflammatory responses. So this is a dynamic process I want to discuss with you about. This is what we're actively studying. So usually when there is a pathogen coming into the mucosal barrier, it's the antigen-presenting cells that will sense them. So these antigen-presenting cells will go to the draining lymph node in the mesenteric lymph node and tell someone. They tell CD4 T cells, hey, there are these pathogens I found. And CD4 T cells realize that, oh, I know which one that is. It's either intracellular or extracellular, and they will upregulate migratory signals such as alpha-4 beta-7, and then alpha-4 beta-7 will be binding to this MACAM at the epithelial endothelial cells in the gut. Basically, these cells will deport and then go into the mucosal surface. At the mucosa, antigen-presenting cells will tell these CD4 T cells again through antigen presentation and cytokine, the local environment, telling that, so these are the pathogens I see, please come and kill them. So because of this is a dynamic process that we found very interesting that we want to study. So with that, our hypothesis is HIV takes advantage of these T cell homeostasis to persist, and they leverage these T cell polarization, proliferation, and migration programs. And currently, we want to understand this. The problem is, if you're a mouse immunologist, you have so many tools you can use to study this. You can use GFP, you can use different genetically modified mice, you can knock in or knock out, you can track, 45.1.2 kind of different, like these adoptive transfers. But for human, such as HIV infection model, you really can't do that. So we want to study the polarization, proliferation, and migration of T cells in humans. How can we do this? Easy. It sounds like we can just single cell them. Yeah, I get all these. You can look at proliferation and migration by what method? Easy. It's just the T cell receptor sequencing we've been seeing. If you see different cells at different anatomical locations having the T cell receptor sequence, so then you know that, then these are different T cells, but they are coming from the same T cell clone that migrated between these two compartments. So that with that, we collaborated with Dr. Luis Montana at with our institute. We collected paired blood and gut samples from people having HIV, all under suppressive antiretroviral therapy. We took their colon biopsy, unfortunately not intestine, but colon biopsies, and look at their gut cell phenotypes so that we can identify all of these different gut immune cells people having different interests in, but we're focusing on CD4 T cells. For example, we can see the different polarization, proliferation, and HIV infection in these CD4 T cells that we pull out in these data set, so that we can easily see that, do you think blood and gut are the same in the immune cell composition? Because people believe that, blood is a circulation of the gut, so you can sequence blood to reflect the gut. This is what Michelle Newson's said, but really, you can easily find things that look different. For example, you will see in the blood you have naive cells, but the gut you don't. And you can also see that there's this population called cytotoxic CD4 T cells in the blood that you don't see them in the gut. Because if you think about this, the lymph node is the home and education center of T lymphocytes. The blood is I-95 or the highway, and the gut is the actual battlefield or the workplace that these T cells will go, so that you can see that only those effectors will go to the gut. And if you have those making granzyme B, you don't want them to come to the gut because that will destroy your gut epithelial cell. So with that, we found the differences between the blood and gut T cell component. We found that HIV-infected cells in the blood are in Th1 or Th17, can be enriched in one or the other. For example, HIV-infected cells in the gut is enriched in Th17. So if we only do blood studies, we will not find this. Now we find that in the gut, they're actually different. And these cells do upregulate IL-23 receptor, which is a Th17 marker. And then finally, we found that those proliferative cells in the blood is cytotoxic CD4 T cells, as we saw before, but in the gut, they're mostly Th1s. So the next question is, people ask us, in these blood and gut clones, do they look same or different? The answer is the most proliferative cells in the blood are mostly cytotoxic CD4s, but in the gut, they're quite diverse, depending on where the biopsy happens and what these cells are experiencing at the time. So even if you find these CD4 T cell clones that are having the exact same T cell receptor sequences between the blood and gut, they may look like one way in the blood, but look at the other in the gut, that they may differentiate based on the second priming in the gut. And we find this very interesting. So finally, now we love to study HIV or the immune cells in the context of tissues. Yes, the way we did this is, you take tissues, grind them up, and sequence them. Yes, we did. But you missed the interaction part. what if this infected cell has a CD8 T cells next to it? And this is extremely important in cancer microenvironment because these cell interactions and pathways dictating these interactions are extremely important for cancer. So there are commercially available methods to look at these cell transcriptomes at the tissue, such as VCM. This is a 10x commercialized platform. But the initial version is looking at 55 microns, which is just a mixture of cells, which doesn't really get us the resolution of single cell. So this is developed by Rongfan at Yale, trying to narrow it down to 10 micron resolution, which is way better, closer to single cells. But there are new methods. This is developed by Junhee Lee's group at University of Michigan using SeqScope, which can look at these cells at the 0.6 micron resolution. So this is authentic single cells. Because if you think about this, these grids, you think cells are squares and squares sitting exactly next to each other? The answer is no. Each grid, you will have maybe 2-3 cells, having part of it. But then now, if you can reach 0.6 micron resolution, you're really looking at the authentic single cells. And finally, this is the method that our lab is currently trying called OpenST. So basically, the trick is SeqScope, you take MySeq chip. We already finished doing that for 2 years. MySeq chips that you typically use to sequence. You crack it open, put your tissue on, sequence it to reach 0.6 micron resolution. OpenST is a revised version using the NovaSeq chips, about this big. So because MySeq chip is small, now NovaSeq chip is big, so you can crack open the whole thing and put the whole tissue on it, and you can even reconstruct spatial transcriptomics at the 3D level. So we found this at bioRxiv December last year. We are already trying this in our lab. I would recommend you to take a look. So in reality, what happened? In reality, what happened is that, we want to study HIV infection in the context of tissues, but we know HIV infected cells are so rare. So we collaborated with Tom Hope at Northwestern that he sent us these tissues of SIV blocks that he knows that these are blocks having SIV signal. Yachi, can you take a look at them? I said, yeah. So we started with lymph nodes. So with lymph nodes, he told us that these are some blocks having SIV. So our student, Kristen, learned to make these chips inside Ron's lab during COVID. So then she is now making these microfluidic devices in her hand in our lab, so that we get these blocks. You cut them to look at H&E to see this is a lymph node. Then you can make sections, so then to put them on these spatial devices, so that you can look at their spatial ATAC-seq and spatial RNA-seq. So with that, she identified SIV DNA positive cells and SIV RNA positive cells, and she's in the process of analyzing these results. As a proof of concept, when we did this in human gut, for example, it's what it works. Here's a gut, then you put your device over so that you can get these these RNA captured. So not only you can see these different clusters of different cells, but also you can look at their spatial organization. So this is a summary of their different fancy spatial transcriptomics devices. And all vendors will try to tell you theirs is, of course, the best. All right. The problem is that it depends on the question that you're looking at. There are two major ways to look at this. One is called genome-wide profiling. Basically, it's either randomer or poly-A-based capture, so that you can capture all the transcriptome-wide RNA expression. The problem is that it's not very specific, because the very top that you're capturing is likely housekeeping genes. Then to read deeper, to reach the specific gene you want, may not be as sensitive. There are also targeted RNA capture. It means that what if you just select these 1,000 genes or about 10,000 genes that you want? Then you can hybridize those, then that will increase the sensitivity. These are either MERFISH, this is by Xiaowei Zhang, or this is Cosmix or Genomics. So this Cosmix is a new version by NanoString. So these can reach much better resolution and much better sensitivity. However, it's a targeted capture. You will have to design your capture in the way that, if there's something totally unknown to the field, such as IKZF3 you never thought about, and if it's not included, you're not going to find it. So it's really depending on what your question is to decide the resolution and tissue size. For example, in these genome-wide methods, starting from Visium, so slight-seq is about 20 micron, and DiviSeq that we tried DiviSeq, C-scope, and OpenST to reach this 0.6 micron resolution that we are currently trying this one because it's the biggest field and the best resolution at the genome-wide scale. I can't promise it because I'm still trying. So if I have results that I reproduced, I hope to share more about this, but then this is Spire Archive and another Belgian group did the same thing. So I suppose people have been doing it. You can also go with targeted capture, such as VCM are advancing their technology. So now you can use targeted capture. But in these ways, the problem is that for us, if we want to capture HIV, those plotted on nanostrings and all things, they don't have HIV. I need to design HIV probes. Otherwise, when I put my tissue on, I will not detect any HIV. So you need to design what you really want to see. Some of the systems are still not very robust, like Xenium, maybe at 1,000 genes. It's not really robust, but like VCM HD, like, and then Cosmics move it up to like about, if it's 16,000 gene captures, like close to 20,000, I would say you can claim genome-wide capture that way. So finally, for people who are interested in single-cell multiomics, so I want to share some of our just some insight that we did pay a lot of money to learn this, but I would say if you're outsiders, you just want to get these beautiful plots and publish a paper. For us, it's just here. Don't be satisfied with these beautiful plots. No, you have data, you have a plot, but you need to see what's really in there to find the biological sense. Don't be satisfied with a beautiful plot you have. And the second thing, what people are worried most about is that this is so expensive and I just don't have the money. I agree. We also don't have that much money. The first question is, do you really need to do single cell? People ask us to say, you know, I have these cell lines, I have this cell line and that cell line, I want to compare them, so I want to do single cell. Hey, if your cell lines are all the same, just do bulk. Single cell is not helping you that way, right? So your first question is that, do you really need to do single cell? Then you think, which platform do you want? If you have known targets you want, let's just try targeted. And then the thing is that how many cells do you need to find the biological significance? For us, you likely need to flow in 1 million cells. Oh my goodness, because we have this 1 in a million problem. But if you're looking at specific populations, such as, you know, tough cells in the gut, how many is that? Is that 1%? So then how many cells you want to sequence to cover that 1% to have, how about a couple thousand for you to develop these biological insight? Then we have ways to hashtag and pull samples. We calculate the costs. We write a minute-by-minute, step-by-step protocol. So this is Changzhi, who was at the seminar 2 years ago, virtually. After the seminar, he emailed me, Yachi, I actually have those like clinical samples in my freezer, but I don't know what to do with them. There's a leukemia. He's a hematologist. I said, oh, well, come, we can show you. So Tuesday, just the morning before I flew off, Tuesday, we did his first single cell experiment together as a hematologist with no single cell experience. He plotted down step-by-step, minute-by-minute protocol practice using cell line model work. Then we put this into the machine, about $20,000 that day, US dollars. I'm hoping that it works. So you can really learn it. It's not that difficult. And what I'm hoping is that once you do the first rounds and you learn how to do it, then your future samples will be way easier. And second, the reason why we analyze single cells bioinformatics inside our lab is like, you know, I have a collaborator, just make him do it, you know. The problem is collaborators will give you plots and you will just take it and publish. The answer is you need to have sanity check that bioinformaticians do not have. For example, if I see these, you know, spatial transcriptomics, my postdoc show me data, I find these genes, I said, do you see CD4s and, you know, B cell markers? He said, no, I didn't. Then there's a problem. If you're taking lymph node, you don't see CD4s, you don't see B cells. I really don't know what's in there. So that's why we do bioinformatics in our lab, because when you analyze the data, you will see exactly whether it makes sense bioinformatics, like biologically, and that's why we need to know. And third, when you have these packages such as batch effect corrections or doublet removals, we read these papers. These are the best practice papers that we read. At least we hope to synchronize. that by reading this, we're now using SCW Finder, and in addition to these, there was so many packages what we use, right? And then you need to understand how these methods work to see which method I should use for my data set. And finally, for gene expression analysis, people typically just do differential expression and get gene ontology, you can do David or Keck, whatever, but it's more than that. The gene set enrichment analysis or WGCNA that can help you the insight, and we use this to find, for example, IKZ of 3 this way. So we routinely send our postdocs and myself included to these meetings. So this is Rahul Satija Single Cell Day. I've been doing this ever since I started the lab, and it's free. So that was this is 2013, but in 2014, the date is March 29th. It's free, it's virtual. We go to every one of them. Just look up Rahul Satija. And then we also go to these cohort and Harvard single cell analysis meetings. I sent all 3 of my postdocs over. They learned so much. For example, Harmony has a new version. You can use that. It's more robust. And there are virtual versions. You can just attend $300 online. So finally, this is what I see where we stand. In the first 13 years when HIV was discovered, just like how we had the SARS pandemic, people try to understand what is it? What is the virus? So we sequence the plasma virus. We try to understand what this virus is. Then, so Bob Sicano and Tony Fauci and Doug Richmond, 1997, they learned that there is a reservoir in people. So then they try to quantify how many, so then we can design clinical strategies. So that was a time where we're trying to measure this HIV proviral landscape. This is viral landscape, proviral landscape. But because of the development of these single cell technologies, so that right now I am no longer satisfied with these methods. For example, when I started my lab, if I did exactly what I did in my PhD, I should be sequencing full-length proviral landscape all my life. The answer is no, I don't do it anymore because I feel like there are things that are more exciting because I want to understand why and how these cells persist by looking at their cellular landscape. I hope I convince you that these single cell beautiful plots, not showing just these beautiful plots, but we found that IKZF3 may help HIV infected cells to proliferate, and there are drugs that can inhibit them. The serpin-B9 may help these HIV infected cells to resist CD8 T cell killing, and there are also drugs that can try to inhibit this, and we're trying. So finally, unfortunately, I'm not sure that my method will cure any HIV-infected individual. The thing is that we're developing tools to sharpen and to our understanding of HIV when we are daunted by this problem of heterogeneity and rarity. These are methods that can be used in many clinical trials and is being used in many clinical trials that we can try to understand HIV-induced immune dysfunction and HIV persistence over time and space. And finally, I would like to thank the study participants and the lead authors. This is Jack and Yulong, who led these experiments, our collaborators and funding sources. Thank you.
那谢谢雅琪非常精彩和丰富的演讲。那有没有什么问题？让我 comment 来。
中文可以，中文。
棒，非常精彩的演讲。我只蛮好奇那个 Granzyme B 的那个 producing Th1 cell， 就那个我觉得蛮有趣的。我只在想说，我第一个想到说，这到底是一个它的之间的这个 association 到底是一个因还是果？就是说，是因为 activate 的 T 细胞因为它比较不死嘛，因为它有 Granzyme B， 它是杀别人，可是它本身有 shield 的，有那个什么 serpin9。所以说是因为这样子的话，所以最后打到就整个感染完之后杀了结果，最后一定是不会死的留下来。所以这是一个果，还是本身 HIV1 会 induce Granzyme B 不要 prefer？ 所以这个问题。第二个问题就是继续这个问题说，那如果是这样子，我们未来要杀，这样听起来，如果有 show 的 Granzyme B， 不管是 CD8 或 CD4 Cytotoxic CD4 要去杀这些 reservoir 的 T 细胞，理论上来讲 Granzyme B 应该是杀不死，因为它有 shielding。那如果是这样子，是不是要 alternative strategy？ 不知道你的看法。
这是非常好的问题，就是我大概每次给都会被问到一样的问题。So the question is it preferential infection or preferential persistence? And does HIV change this, or is passively following a biological process? The answer is we don't know yet. we have a second-year PhD student who is writing her thesis proposal right now, trying to answer this. So we're trying to use some memory cell models, trying to understand. My guess for now would be, I am guessing, is preferential persistence. That, you know, you have activated CD4 T cells, you can have HIV infection, but then with this in vivo selection pressure, if you have these, you know, cytotoxic responses, that you may persist better. My guess is persistent, but again, we are still testing it. The second question is that how are we going to move this forward to clinical therapeutics? Hold on. So I am not the kind of person who say, hey, let's just remove all cytotoxic CD4 T cells so then people will be cured of HIV. The answer is no. Because you can also remove all CD4 T cells and people can be cured of HIV, but they'll have this iatrogenic AIDS. So no, I'm not the kind of person. But then, how are we going to move this? So I don't know. So there's so many layers of HIV-induced immune resistance. For example, there can downregulate NAF to reduce, your NAF can downregulate MHC presentation. You know, granzyme B is one, you have perforin, you know, you have different fat, and you have different ways of killing. So we don't know. It's still a multifactorial way that we can sensitize some cells for killing, maybe, but hopefully not at the cost of hurting CD8 T cells. That's a major concern. And how to do that, we don't know yet.
谢谢。那还有没有在其他的问题？
Let me try the first. The question is, are the tools sensitive for epigenetics? So the current method I'm showing is only ATAC-seq. How much do you trust us to reflect the epigenetic regulator you're looking at? I'm not sure. For example, if you're looking at CpG methylation, there are other ways. You know, you look at CpG methylation. If you want to look at, you know, histone like conformity marks, there are different ways that we have a different paper on high C, high CYP4C to do this. But what I mean is that the first question is ATAC-seq alone sufficient to answer the epigenetic question you want? This is the first. And second, I prefer not to send PBMC and tissue to me, but send the person to my lab. Yes, here. So here. So we have a hematologist right here. Here it is. Here he is. There he is. Okay. We have a hematologist with no bioinformatic background who can learn to do weight as nearest neighbor in 3 months. 3 months. He already did his first single cell experiment this Tuesday. And we envision that he will learn everything and go back. And we want him to be the best single cell person in Taiwan. And I hope you can all learn this either in my lab or somewhere else. The importance is to learn the tool. So just come to my lab.
好，就是送人过去或自己过去都是好的办法哈。那还有没有其他的问题或 comment？ 是冰冰。
技术性问题，这边其实我很好奇，就是有一群 HIV DNA 那个是你会学友会觉得是安全的问题，因为就是目前就是学友使用的方法就是 DNA HIV DNA 不太这个一开始，会觉得会是一开始的
Yes, it is.
重复一下问题，才会被录进去。
这个问题是，就是我们看到有 DNA positive DNA RNA positive. Why is that? Well, this is a very good question. We actually have it. So here, imagine this: the HIV proviral genome is 10 kb, but the human genome is 6 times 10 to the 9th for diploid. Okay, you can of course argue that there are open and closed chromatin regions, and open regions are probably not that much. But the problem is that we do have sensitivity issues. So we use a cell line model, each having HIV in there, and we measure that this capture efficiency is 28% only. Imagine this difference. That 28% we feel like is already pretty good, and then we read twice deeper is what 10x recommended, trying to compensate for this. But then it's the detection limit rate is probably just 28%. So yes. So if we see HIV in there, we say there's probably authentic HIV. But if we don't see it, we cannot say they're negative, and that's why there will be HIV RNA positive and DNA negative, and because RNA has many copies for you to capture, but DNA only has one copy. And if a transposase is not binding to it, you are not going to see that. And then there is accessibility issue. If that region is not accessible, so you won't capture it. So the answer is yes, there is a detection limit problem. Yeah.
那可能贴身知道它的可能的稀有度，就是相对于说话的那个生体以外的话 ，GAP 的贴身大概是可能稀有。那这个除了 GAP 以外之外，有想要去 explore 其他 tissue present 的 T cell 有不同的 shield mechanism？
It's a very good question. You want to come to my lab? Okay. Here's a question. So the first question is, why do HIV infected cells preferentially enriched in T17 in the gut? And why? The answer is, I don't know. We're studying it today. Now, all right. The second thing is that these methods of mechanisms of persistence, whether it be different tissues. For example, what we're interested in right now is blood, gut, and lymph node, because we know HIV persists in these major sites, and how they persist in lymph node, I believe, will be quite different from how they persist in the gut, because gut is a workplace, you look like this, but lymph node is a home, you look like that. So they do have a different mechanism, but we don't know yet. Send someone, interesting students.
好，还是有一些好人要留在台湾呐，就是好。那再来，还有没有其他的 comment 或问题？我问一个哈，就是你如果做 TCR 的 sequencing， 你就是看 CDR3 而已，而不是全场，然后目前的技术上
Yeah, CDR3 region.
OK， 好。
So it's based on different publications. The CDR3 region is likely sufficient. You look at the VDA junction. The junction site is predicting their sequence specificity, supposedly.
OK， 尤其是这个是的确是一个 trade off， 但是全场至少是 TCR 的话，它反正没有比较多的 somatic mutation， 所以 CDR3 probably is sufficient to predict the whole sequence. Okay.
It's just like the same method can be used for B cell receptor sequencing, for example. And then so the thing is that B cell diversity is driven by somatic hypermutation, but in like a T cell difference it's by the recombination.
Yes.
It's not by somatic. So that one is sufficient to by other studies sufficient to tell specificity.
还有没有其他的问题？好，线上是。那当然我们的好处就是这个是会变成一个在台大演讲网的一个 video， 所以今天的现场或线上的听众，或者是你是未来听到的都 OK。那雅琪总是3年内、5年内、20年内都可以接受大家的 consultation。好，那现在的问题是什么？
So the current question is, for pathway analysis such as GSCA, do you find any differential expression genes or pathways in these latent or HIV infected cells? So yes, we did find some. I think in both of the studies, I think we put them somewhere in the manuscript. Yes, there's some cytotoxic T cell genes, interferon response genes, TNF pathways. I think TNF pathway is one of those that are enriched. So there are 3 ways to look at, expression profiles. One is differential gene expression, and as you see a volcano plot. This is gene state enrichment, which means that typically you start with those M6DB collections. Basically, people publish these data, and then there's a place called collecting them called M6DB. So you have the hallmark gene sets or other different gene sets to compare. For example, John Worry's exhaustion markers. Do they look like your exhaustion markers? But then I would say the third way is to use WGCNA to see what's inside your data. You don't compare with John Worry. You come to what's inside your data. That's WGCNA. So I would say there are these 3 ways. We do find things here and there, like IKZF3 we found it, and set other pathways we found. So yeah. So in conclusion, that try different ways of gene expression analysis. You may find different insight.
那非常谢谢大家来参加这个 meeting， 我想大家都学到非常非常多。
谢谢大家，谢谢大家过来。